good to be here, good to be anywhere. And see old friends and new friends. And since I understand that this has been recorded, and I guess there's a transcript that's going to be made, I want to start by the usual disclaimer that all aspects of policies that will be discussed, that I will be discussing, are actually my views and don't represent the views of the Department of Energy or the U.S. government. So I think that needs to be made clear in the beginning. This is not a type of talk that I usually give. So in a sense, it's new territory for me. And it's about an issue that I have to make an a priori confession, dealing with publication policies and sharing of data policies that, as I say, it's not something that had occupied me as a practicing scientist, and also had not occupied me at all during the early years of my career as a research management. I have to say that some of the first inklings that the issues that I will address today were important enough actually started when I was involved in the Global Change Program in a significant way before I got involved in biology, and Charlie Kennel and I were colleagues, in fact, at the time. And I became acutely aware at the time that issues dealing with data sharing in global environmental change, in global climate change, for example, were extremely important for the success of the endeavor. First of all, because the problem that was examined at the time, the problem that we were studying, was in fact global in nature. And the notion that you somehow could be successful without the sharing of data at the international level was ludicrous. I also was fortunate enough during my career as a program manager in the Global Change Program that also Charlie was in, that I was sent to Geneva a couple of times to be part of a U.S. delegation that was negotiating, in a sense, the policy that we referred to at that time as the full and open exchange of data. In essence, across boundaries, the sharing of information and data at the cost of reproduction of the data. And it saddens me to some extent to see that on that front, internationally, we've really regressed, primarily because some of the observing systems in other parts of the world have actually deteriorated. But worse yet, some of those data that we always felt were extremely useful just for scientific purposes have now triggered some private sector interests in weather information or climate information, and there is a tendency, at least internationally, not to hoard data and to only make it available for profit. And that's really a setback. This was one of the first inklings. I said that these were issues that were not just for those data weenies that worked away in some corner, but they actually could make or break the success of the scientific enterprise, whether it was in environmental studies or eventually in biology, which is really where I will be spending most of my time. Also, when it came to the success of scientific enterprise, and that's also something that Charlie Kennel knows very well, when NASA took on a fairly significant role in global environmental change and devised the Earth observing system, the constellation of satellites to study the Earth from space, I recall, and I'm sure he and Walter may recall also, how EOS-DIS became a sort of make or break success point for the Earth observing system and also for the Global Change Program per se. So a few years later now, when the Human Genome Project was gearing up for the large-scale sequencing efforts, I was part of the so-called Bermuda Group. It was now famous, or depending on our point of view, infamous, that set of meetings that in some way hammered what's referred to as the so-called Bermuda Principles for the sharing of genome sequence data. Not long after that, we also got caught up in the genome sequencing race between Celera and the International Human Genome Sequencing Consortium. And there was much informed, I would say, and uninformed discussion about issues such as BIDOL, which became law in 1984, about intellectual property, about patents, and the like. Eventually, we also came to February of 2001 with the publication of the 2 papers in Science on the human genome sequence and Nature, with all the controversy that was associated with that publication that I will also address in some detail. Another important event in a series of events that, in a sense, shaped some of my thinking, but also shaped some of the issues that I'll be touching on today, is also September 11 and the anthrax attacks, because it raised issues about sensitive but not classified data. And that's something I'll deal with a little bit. Now, the question comes up, why would I give a talk like this, which is the first for me in some way, in a topic that at least until very recently I didn't know very much about, and about which there are probably individuals, maybe even in this room, that know about these topics a lot more than I do. Why am I doing this? I think it's very, very important, personally, that more scientists weigh in on these issues. Alternatively, many of the decisions that will be made, and decisions will be made anyway, whether they're by default or by interaction and initiative, they will be made in a more ad hoc manner and by individuals who, in my view, have little appreciation of the dynamics and the vulnerabilities of the scientific enterprise. I think there are probably too many cooks that are stirring the broth in terms of data sharing issues. There are funding agencies, there are, of course, journals, primarily through their editors, but also through the publishers. There's professional societies, there are non-governmental organizations, NGOs, there are, of course, private companies. There's Congress, and there's the courts. Again, much is at stake as far as I know. And one just example that many of you may be familiar with, but if you're not familiar with, then I want to awaken you about. And that's an issue that I think is a huge threat to the research enterprise, and that's the current threat we're facing to the research exemption that's enjoyed by universities. That ends almost a recent rule by the Court of Appeals has ended a 170 years old practice. This is a federal appeals court that reversed a decision by a lower court that, in a sense, affirmed the research exemption. It reversed it this past October, noting that the institution that, in fact, was the subject of this legal tangle is, in fact, a business-like entity that profited from the use of, in this case, the lasers. The research unmistakably furthered the university's legitimate business objectives, including educating and enlightening students and faculty, and helped it, and this is the part that I find very offensive, lure lucrative research grants. This was written by the Federal Circuit of Appeals Judge Arthur Gajarsa. This, no question, is going to throw a serious wet blanket on a lot of research at universities if university scientists have to start worrying about, in a sense, violating some of the patent agreements, intellectual property issues. So to finish my introductory remarks, why am I doing this? I'm telling you as a bureaucrat, because that's really what I am. Don't leave it up to the bureaucrats or entirely to the political process. I think university scientists like you have to get very involved in this process. So the three topics that I want to discuss today, And I certainly prefer that this be a discussion as opposed to just a presentation with questions and answers at the end. So if any one of you wants to interrupt me during the course of the discussion, please do. So the first area I want to touch on deals with publication policies by scientific journals, and especially with respect to the availability of the supporting underlying data. And in fact, Dan Drell, a colleague of mine, and I published a short commentary on the subject in Nature that showed up last year. The second area I want to address deals with data release timing and conditions, with particular focus on sequence data, although it probably has application on other data as well. I will stress mostly pre-publication data because that's a real hot topic these days. And the third area is one that I already mentioned and recently received a lot of attention, the one dealing with possible restrictions on data that are relevant to bioterrorism or anti-bioterrorism research. Again, much of what I have to say is relevant to other kinds of data. I've already made some reference to the climate change data, although my focus is mostly on biological data. So let's start with the publication policies by scientific journals. And I've already mentioned the celebrated case, which is, of course, the publication of the working draft of the human genome sequence in February of 2001 with the International Human Genome Sequencing Consortium publishing in Nature, and Celera Genomics, the private company that presumably raced us for completion of the working draft in Science magazine. I've already mentioned that the International Human Genome Sequencing Consortium followed the so-called Bermuda Principles, depositing, in a sense, their raw data, the raw sequences and the assembly data almost overnight in GenBank. And just reading from the Bermuda Principles, I'll read you a few statements because they're kind of important. These were agreed upon on February 96 in Bermuda, and they were endorsed by all parties in 97. And as much as we rejoice and celebrate those principles, I mean, you have to realize that these are not legal agreements. They're more agreements among gentlemen and gentlewomen. So they're never written as legal documents. And as a result, there's quite a bit of ambiguity in how people have interpreted those principles and how have they applied them in their practices with respect to sequence data. So anyway, let me read you just a few parts because they're relevant. So primary genomic sequence should be in the public domain. This is one of the principles. And the other one is primary genomic sequence should be rapidly released. And I read some more. Sequence assemblies should be released as soon as possible. In some centers, assemblies of greater than 1 kb, 1,000 base pairs, would be released automatically on a daily basis. Finished annotated sequence should be submitted immediately to the public databases. And this is an interesting part. It says it was agreed that principles should apply for all human genomic sequence generated by large-scale sequencing centers funded for the public good in order to prevent such centers establishing a privileged position in the exploitation and control of human sequence information. Note that reference was only made, in fact, to human genome sequence and silent for all others. And as you'll see later, that is really an important issue that needs more clarification. And just for the sake of completeness, those principles were in fact endorsed by all participants, and they included in the UK the Wellcome Trust and the UK Medical Research Council. In the US, the NIH National, at that time, Center for Human Genome Research, later becoming the National Human Genome Research Institute. Our office in the Department of Energy, the Biological and Environmental Research Program, the German Human Genome Program, the European Commission, HUGO, which is the International Human Genome Organization, and the Human Genome Project in Japan. So, I also want to make reference to a very thoughtful OECD report that came out last year, and in fact had some folks from your university here involved. that was relevant both for the sequence data as described, but also for the sharing of pre-publication sequence data, a topic that will be discussed later. This was an OECD follow-up group report on issues of access to publicly funded research, and it had some pretty well thought out recommendations and analyses that are worth mentioning. Now, to get back to the issue of publication by Science magazine of the Solera paper. As most of you know, or some of you know, Science magazine and its chief editor were in fact pilloried by some members of the community for placing restrictions, in this case any restrictions were found objectionable, on the data underlying the paper. Even though it's important to note that the data were in fact made available, and let me just summarize without getting into great detail. The data were made available principally through the Solera webpage with different conditions for academic scientists and private sector entities, as well as with some restrictions on the amount of data that could be downloaded at any one time. So there was a lot of concern by members in the scientific community that what science was doing was a dangerous precedent that would create a slippery slope of more and more restrictions on data underlying the scientific process, the scientific papers. So there was also concern that the so-called long-term commitment to full and open access would gradually erode. And in fact, the Academy set up a study section with Tom Check as the chairman, and I think the report that they are supposed to be working on is pending, and I think about to come out at any time, and I'm actually waiting anxiously to see what they have to say. Just for further information, in April of 2002, last year, Science magazine published 2 papers reporting on the draft genome sequences of 2 subspecies of rice. And one of those was, in fact, from a company, Syngenta, that had limitations similar to Solera's limitations for the human genome sequence. Now, lest people think that this was, in fact, a precedent setting both the Solera paper and the Syngenta paper. In fact, it isn't quite true, because a few years back, in fact, in 1987, there was a sea level rise paper that was published in Science magazine by Bilal et al. And they were, in fact, scientists at Exxon. And the title was Chronology of Fluctuating Sea Levels Since the Triassic. And there was it was published in the March 6 issue of Science magazine of 1987, and there was also a news report by Dick Carr from Science. This was also a paper where some seismic data were in fact not released because they were proprietary. They were owned by Exxon, and of course their interest in searching for oil was obviously in their minds compromised if they were to share that data. Nevertheless, Science did publish that paper, and those data are still not made available that underlie the paper. So there was another precedent that was set. At the meeting that we held at the Academy, Eric Lander, who was one of the big, and is one of the big champions of full and open access, said, and I quote, "If you choose to publish a claim, you must release all the integral data supporting it as determined by editors and peer reviewers." As we said in our article in Nature, we felt that it was nevertheless worthwhile to find ways to encourage publications from private sector scientists when the demands for unlimited access to the underlying data is unacceptable to their corporate masters. So we thought about different ways by which we could somehow find a middle road, still believing that nothing is ever absolute, and that negotiation, and what I used to call at the time fighting a battle one trench at a time, was preferable to falling on one sword in the beginning of the battle. So one suggestion we had was to start a timer on the deposition of the data that was at least initially restricted and for some time, and entrusting the deposition of the data ultimately, when that timer ran out, to a trustee that was acceptable to all sides. In a sense, it's similar to how the Patent and Trademark Office allows up to 1 year before a provisional patent application is converted to a utility patent application. So maybe a year could be a reasonable compromise, or at least a suggestion, a proposal at this time. And we thought about different trustees, and something like FASEB, which is the Federation of American Societies for Experimental Biology, could serve as such a trustee. Needless to say, regardless of what we would propose, some pilot studies would need to be done so that some of those ideas could be explored. For example, a pilot study that examines how much a company is helped by restricting access or damaged by more widely sharing data. Those kind of pilot studies would be enlightening. We felt all along that we needed to provide strong incentives to the private sector to avoid the hoarding of large amounts of data that may be of limited value from the commercial point of view, but of significant value from the basic research point of view. We felt that universities are the best go-betweens by offering adjunct appointments, for example, to private sector scientists who could provide legitimacy and academic status to work done by private sector scientists. And my colleague, David Gallus, actually enlightened me with respect to something in his institute, the Keck Graduate Institute, that has graduate students that are conducting research that is sponsored by private companies under confidentiality arrangements in exchange for publication rights for the students that study there. There were other also precedents for public-private cooperation that could be explored as models, such as the SNP Consortium, the Single Nucleotide Polymorphism Consortium, and the IMAGE Consortium. Both are sort of win-win arrangements of public and private entities. And both placed valuable genomic sequences in the public domain. There's no question that there are some high risks, some risks, I would say, for private entities, such as corporate takeover and disbanding, such as the one that happened with Research Genetics, that is a distributor of a set of clone resources. So we concluded in our commentary that whatever policies are promulgated by scientific communities, it's really the journals that ultimately will have to enforce them. We felt that trade-offs had always been between the prestige of publishing in Science or Nature in exchange for openness and unrestricted access to the relevant data. So again, to say, we wanted to find creative ways to work this problem, the increasing fraction of 60% of genomic research that's conducted in the private sector will remain unavailable otherwise to academic and other scientists. And we think it's too high a price to pay. So we do need to create incentives for private sector companies to publish data without proprietary constraints, rather than clamoring for policies that push them towards non-disclosure. Now, I want to make it very clear that there is no question that full and unrestricted access to fundamental research data should remain the guiding star of most scientific research, because frankly, centuries of experience suggest that this is the best way to achieve progress. And I don't have the quote with me, but I actually even found one attributed to Aristotle, which is, a couple of thousand years old, that basically said the same thing. However, having said this, we must accept current realities. Private sector science has its legitimate interests and requires, therefore, openness to new approaches on our part, collectively, bereft of fundamentalism regarding access to data that governments did not fund and cannot claim to own. Another recent example that I want to make reference to is the publication by Celera in Science of mouse chromosome 16, I think, which wouldn't have happened had there not been some flexibility in making those data available in other than the Bermuda Principle concept. So now let me shift to the second topic, the sharing of pre-publication data, and again, specifically genomic sequence data, where I could make a more sensational title by saying, will Bermuda Principles rule the future, or will we have some other alternatives? The issue of the Bermuda Principles has actually heated up considerably in recent months. I have here some notes about Bermuda, but I'll skip them. It's pretty much what I told you earlier when I read the except in greater detail. But you can get to the web page and read some of the details. I have to say that in the early days of some of the sequencing projects, especially mostly on the human genome sequence, the adhering to the Bermuda Principles was mostly symbolic, with one significant exception. And again, the exception, of course, was Celera, which downloaded the public data and used them in combination with their data to come up with the assembly of the human genome sequence working draft. And the extent to which they used them, is really still subject to some controversy and debate. There are some folks that claim that they actually did use the assembly that the public program provided, whereas Celera claimed that they actually shredded the data and used them in the assembly that they put together for their working draft. But it's not an issue here, and it's now been discussed in the pages of PNAS. It is true that back then, bioinformaticists, however you define them, were actually not poised to pounce on the data as they became available. I would say, the last year, times have changed considerably, and there are, in fact, folks, bioinformaticists out there, that are, in fact, ready to pounce on the data that become available. They're primed and ready. And in fact, the impersonality of the web makes some of that pouncing and using much easier. There is a recent example that many of you know of, the experience of Lee Rowan and et al., who were scooped, and their problem was, in fact, discussed in the pages of Science. Let me give you a few details of this for those of you that don't recall it. This was the Rowan, Wong, Lane, and Hood work. They did the sequencing and deposited the data dutifully, like most scientists do in the public databases. This dealt mostly with 1.5 million of annotated sequence from the mouse major histocompatibility complex. During that time, there were two papers that were submitted to Immunology Today by Alcock et al. And Yu et al., published July 1 of 2000. And Elaine Bell, who's the editor of Immunology Today, accepted the paper, even though they were based on data that Rowan et al. Had actually worked on and had not in any way given permission for the others to use. However, the editor of Immunology Today claimed that there was really no policy governing the use of publicly available data, which is true. That sufficient time had elapsed since the data had been deposited. In fact, it was over a year that the data had been deposited. And there was an offer of co-authorship made to the four scientists, which was not accepted by them. Rowan et al. Responded, basically saying that the most important point they were making was not addressed when they argued the case. And that's that the actual sequencing projects and the data on which those papers were based were in fact not finished. And I'll read you some of the position of Rowan et al. We do not believe, they said, that reviews should be written on unpublished data and analysis that have not gone through the peer review process. They also said, we do not sanction the preempting of obvious primary publications by third parties when permission to do so has been neither sought nor granted. And another point they made, they said, projects such as this one are of sufficient complexity and the time frame for mapping, sequencing, and analyzing the data can be long, especially if funding resources are limited. So what Rowan et al., in fact, very, I would say, in a conciliatory way proposed was that all deposited data as a way to solve this problem be also tagged. So the tag would indicate to some extent that the submitter requires permission for publication of analyses based directly on the sequence that's been deposited. So one of the proposals, despite the fact that they got burned, they figured, well, you know, there really was no rule that was violated, and it was more the etiquette, the scientific etiquette that was violated. So they offered this alternative approach, at least for the future, that all the data could be made available fully and openly, but for those data where the principal investigators had some grand plans in terms of further analyses and publication, the data should be tagged, and maybe a small fraction of those data would have been tagged. And in those cases where outsiders would access the data, if they would see the tag, they would have at least the obligation to contact the PIs and explore possible partnership in the representation of the results. Driven, I wouldn't say primarily, but to some extent by the Rowan et al. Episode, the Bermuda crowd, the folks, many of us who were involved in struggling with this issue, met quite recently to take up this task. And we were bound by the so-called Chatham House rules that Wellcome Trust instituted, so I can't give you many of the details, but I'm free to give you my opinions, which I will do. And I confess that I'm somewhat conflicted on this issue of sharing of pre-publication data. And in fact, you know, even if you take it one step further beyond data to other resources like reagents and clones and others. So let me see, what are the advantages of quick and unrestricted access to all data upon production? And again, if for sequence data, the Bermuda Principles of 1,000 kb, 1 kb fragments, and full assemblies when completed. So I was trying to think what would be the advantages here. First of all, I would say, well, early and unrestricted access is the democratic thing with a small d, particularly for large community-based projects. And one can argue what's the definition of a large or community-based pending is probably something that most of you know what it is, but it's still to be defined because there may be projects that are on the border. So that's one obvious advantage. And I mean to specify that democratic with a small d is in some sense that the centers that are well-funded to produce sequence data don't have too much of an unfair advantage of using the data for high-impact publications. And that's a democratic argument that one can make. There's also no question that improved competition with a net result is a good thing, and it will have the net result that publishing occurs quickly and discoveries are out there also very quickly. It's like Solera's slogan that discovery can't wait. Also, I would say that journal editors, funding agencies, other policy types are not called upon to adjudicate disputes between scientists complaining about being scooped by other scientists in the rush to publish. Basically, decisions are entirely based on the quality of the scientific arguments. So In a way, it's a cut and dry. Whatever data you make out available, anybody can use it. You don't have to answer to anybody. It's sort of the law of the jungle. Whoever is fastest can do it. And since it's so simple, there aren't too many cases to adjudicate. If the data is out there and it's available, you can use it. You don't have to answer to anybody. So what are some of the disadvantages? Obviously, we're going to have sloppier scientific publications because results will more often have to be retracted. There's going to be destructive competition. You know, there's going to be a breakdown of civility and noblesse oblige. There's also going to be, no question, a reluctance on the part of some big centers to take on the community task of generating resources that they don't have the opportunity to take first crack at. And also, what I said earlier as an advantage could also be a disadvantage, and that was, in essence, journal editors or funding agencies, you know, don't have to adjudicate. So that could also be seen as a disadvantage. I expect that most funding agencies will take a position on this rather soon. I know the welcome is rushing to a decision. I know our colleagues at National Human Genome Research Institute want to bring up this issue at the next council meeting, which is coming very soon. So at least for large genome sequence data, this issue will probably see some resolution, or at least some consensus, reasonably soon. I like Tim Hubbard's sense of finding a Creative Commons solution. I like that term very much. This issue actually is of particular importance to us because in the Department of Energy, and especially in our production genomics facility in Walnut Creek, part of the Joint Genome Institute's operation, we are focusing primarily on microbial sequences. And whereas what I've said already probably deals with the larger genomes, when it comes to microbial sequences, the situation is even more messy. We haven't yet formulated a policy about the microbial genome sequences, how they're made available, how quickly they're made available, what agreements need to be struck in terms of people having access to the data. We're still searching for some resolution. There is a sense in the microbial sequencing community that microbial sequences should not be shared until at least you have a 3x or 4x coverage and you get a first-order assembly for the genome of the microbe. Like with the previous case, there is even a bigger concern that data will be scooped by outside scientists who will rush to publish, thus scooping the scientists principally involved in the study. And this has actually happened in some cases. And for example, the TIGER, the Institute for Genomic Research, which keeps all their data on their web page and doesn't submit that to GenBank, feels that they had been scooped in this particular case. And in fact, they are withdrawing a little bit from some of their more open policies. Although this issue is still, as I say, at an impasse and needs to be deliberated and adjudicated and some reasonable solution found. This is a case where many more agencies and many more players are involved, not just the few labs, the few entities that were involved in the human genome sequencing. And it is also important to state that GenBank is refusing any kind of compromise in terms of restrictions and is accepting no other solution than the complete and totally unrestricted access to all the data. I would say that the issue gets even more complicated as we consider other types of data. For example, when we've heard from the structural genomics folks where the period of deposition can be as long as a year up until very recently. And I think the folks at NIH, General Medical Sciences, are contemplating narrowing this even down to 2 weeks. I don't know how that could be done. I know that NIGMS is considering this community aspect of the structural genomics program and demanding this more rapid deposition of the data in the protein data bank. So when the program gets humming, I suspect that this is an issue that's going to be dealt with So let me move now to the third area, and that's the national security and biological research data. I mentioned that the situation became particularly acute post-September 11 and the creation of the Department of Homeland Security. This is a case where national security is pitted against traditional openness of scientific research. Many of us who've been in this business can recall the situation during the Cold War. Although even at the height of the war, in 1985, the National Academy of Sciences concluded that greater security would be achieved by open pursuit of scientific knowledge than by curtailing free exchange of scientific information. That was, in fact, codified by the Reagan administration through the National Security Decision Directive 189 that states, and I quote, "It is the policy of the administration that to the maximum extent possible, the products of fundamental research remain unrestricted. It is also the policy of this administration that where the national security requires control, the mechanism for control of information generated during federally funded fundamental research in science, technology, and engineering at colleges, universities, and laboratories is classification." The Clinton administration in '95 also issued an executive order that stated that basic scientific information not clearly related to national security may not be classified. So we've had sort of a long history of how to deal with this issue, although I must say this has been primarily in physics, in chemistry, and engineering. Since the events of September 11, we have had some conflicting messages from the administration, and it may be just a matter of clarification, and we will get that hopefully very soon. There was a letter by Condoleezza Rice, the President's National Security Advisor, in a letter to Harold Brown dated November 2001 that affirmed the importance of openness in fundamental research. In fact, it said, "The key to maintaining U.S. technological preeminence is to encourage open and collaborative basic research." It also said, "The policy in the National Security Directive 189 shall remain in effect." On the other hand, there was a memo from the President's Chief of Staff dated March 19 of last year to the various agencies, telling them to take steps to protect information regarding weapons of mass destruction. So the issue that's raised here, so the specter that's concerned some people, is this business of non-classified but sensitive information and where you draw the line. I think that concern, or not just I, but others feel that concern about sensitive biological information grew primarily from a couple of papers. One that was published that showed the insertion of the IL-4 genes into mousepox viruses that resulted in almost total immunosuppression. An important point to note in this particular work was that, in fact, it was done in Australia, so whatever rules we would have instituted probably wouldn't have been applicable here. The second paper was published actually demonstrating that an artificially synthesized poliovirus genome produced an infective pathogenic virus. So there was obvious concern that such information falling into the wrong hands would create serious problems, would give, in essence, potential terrorists information and knowledge that we didn't want them to have. Over the last year, there have been many, many discussions on this topic. There have been hearings, there have been National Academy of Science meetings. I know my colleague David Gallus has invested time and effort in some of those meetings and others. In essence, bringing together two communities that, in fact, have often seen each other with great distrust, and that's the security experts and biologists. John Hamre, who was in fact the former Deputy Defense Secretary and now is President for the Center for Strategic and International Studies, cautioned many of the people involved in several of those discussions that if we don't start now in terms of coming up with a constructive dialogue with each other, we're going to turn this into a disaster. That was his quote. It is true that these issues are not new. just like I mentioned Aristotle a little earlier, it turns out that in 1626, Sir Francis Bacon, who is credited for establishing the scientific method — I think the scientific method was established personally much earlier, but I'll leave it — concluded that there were times when secrecy was appropriate. And of course, we know about the Manhattan Project that I made reference to, where there was definitely a culture of secrecy in physics, chemistry, and engineering. The same thing happened with computer scientists and mathematicians in studies dealing with cryptography. But this situation is rather new, I would say, for the biomedical sciences. Although I remind some of you about the recombinant DNA discovery that prompted the famous Asilomar workshop back in 1975, where the leading scientists worked openly to propose ways to manage the risk. Out of that workshop came the NIH Recombinant Advisory Committee, the RAC as it's known, with its decentralized institutional biosafety commissions that are now also used, that concept, by other countries in dealing with things such as the creation of genetically modified organisms. The recent deliberations have in fact focused on 64 biothreat agents, and some have contemplated restricting work or information dissemination on those 64 biothreat agents in all the associated publications. It's important to note that contrasted with the Manhattan Project publications and research, there are some differences here with respect to microbiology. And there's no question that microbiology now is extremely international. A very large percentage of the authors of the papers in microbiology are, in fact, from overseas, and the same thing for the reviewers as well. A concern that we also have is that this anxiety and some of the ideas that are sort of promulgated will contribute to xenophobia, and in some way restricting or discouraging graduate students from overseas in coming to our universities and working in our problems. I believe at least, that the wars against terrorism and infectious diseases are in fact global. If government moves towards restraining the flow of information across national boundaries, there will be an inevitable clash with the academic research community that is increasingly seeking international collaborations and partnerships. There's no question in my mind that it would impact biomedical research and delay these very cures that may in fact be what we need to effectively respond to a bioterrorism attack. In terms of getting a handle on the problem, it's interesting to note that the American Society for Microbiology, ASM, publishes about 6,000 papers per year. And during this past year, only a handful, in fact, raised concerns among the editors and the reviewers about the information that was in these papers. In fact, no paper was turned down by any journal, by any reviewer, by any editor. Nevertheless, for some papers, very few, I think it was like 5 in total, the interaction or the interplay between the author, the reviewer, and the editor prompted the author to withdraw the papers. The authors to withdraw the papers because they felt that in some way, they may have provided information that was inappropriately, that would have been inappropriate to share. I think there is a general consensus in this particular case that what we need is to identify precisely the specific areas that require classification, and build high walls around those. So it's pretty much the same concept, the same approach that we've used in the classification process that was in effect during the Cold War and the Manhattan Project. Now, what are some of the examples of information where you would like to build high walls around, where you would seek classification? And some of those that I've in discussing with colleagues I've picked up were such things as sequences for particular stretches of sequence, for example, that could be used to identify individual pathogens, in a sense showing information about detectors that would be used to pick up signals or the presence, or to detect the presence of a biothreat agent. And under no circumstances do we think that sequences for non-pathogens should ever be restricted. I think this is a very dynamic process, and hopefully, again, like with the previous two cases that I described, we will get resolution in a reasonable amount of time. And that resolution will be one that will protect us from potential abuse of this information by terrorists, while at the same time not creating any impediments or any restrictions to the full and more vigorous and vibrant applications of the research enterprise. So I will conclude with one of the things I said in the beginning, and which is the reason why I'm giving presentations that in some sense are pretty boring, but they are actually quite important. I think that scientists, most of you, need to be engaged in this process, and I urge you all not to leave this issue just to the bureaucrats like myself. Thank you very much.